This was a study conducted in MDS unit in Florence with the group of Professor Valeria Santini, and we retained that the best approach to study de novo younger MDS patients is to perform an high-throughput sequencing techniques, so an integrated approach in order to define the molecular and cytogenetics characteristic of these patients and to define the frequency of germline predisposition frequency that could justify this early onset. We also performed in these patients, in this cohort, low coverage whole genome sequencing on plasma cell-free DNA. That is an assay conducted through the peripheral blood, so it is a very invasive study in order to define some cytogenetic alteration. And so we studied 31 patients affected by de novo MDS, and we surprisingly noticed that at least 50% of these patients presented a germline predisposition that we reconducted in 4 different groups, in particular DNA repair defects and cancer predisposing variants, the predisposition to myeloid neoplasm, inherited bone marrow failure syndromes that we used to see in pediatric patients, and also variants related to hereditary anemia, so for example red blood cell defects. And so we noticed the importance of a complex mode of inheritance regarding some patients, and also some atypical genes not previously reported in MDS cases could be more informative in predisposition of MDS patients.